Okay, so this is my updated version of my eternal battle of good versus evil and the process of mitosis versus meiosis. You know, every year I teach this lesson and students will sometimes mix up a few stages, so I hope this video will help clarify. So what the picture shows is a cell going through the process of mitosis. A human cell has 46 chromosomes. That is what we call the diploid chromosome number. And in the process of mitosis, a diploid cell with 46 chromosomes will divide into two cells, each of them also containing 46 chromosomes. Well, that's a little different when we compare and contrast with meiosis. In the picture, we see a cell starts very similar when it goes through the process of meiosis. A human cell, will start with 46 chromosomes, but through the process of meiosis, that 46 chromosome cell will divide not once, but twice into a total of 4 cells. And each of the 4 cells has half the total number of chromosomes. That's what we call the term haploid. Haploid cells have half the total number of chromosomes, and in humans, half of 46 is 23. So how does this process take place? What are the stages of similarities and differences between mitosis and meiosis? That's what this video is all about. Let's go ahead and get started. So here we have two cells, and the cell on the top will eventually go through mitosis, and the cell on the bottom will eventually go through meiosis. Right now, both of these cells are in interphase. Interphase takes place prior to mitosis, and interphase takes place prior to meiosis. And what I want to do is first count out the amount of chromatin, DNA, strands of DNA that are in the nucleus of these two cells. And so I want to make a note. You know, just to start, in the top cell, we're starting with a cell that has 4 strands of DNA. Well, look at the bottom cell. In the bottom cell, we also see a cell that has 4 strands of DNA. And just so we don't forget that number, I'm going to go ahead and make a note of it in the lower left-hand corner. And that note is going to stay there the whole time as our silent reminder of how much DNA was in each of these two cells at the start. So they each have 4 strands of DNA called chromatin. Well, now that we're going to go through this process, what happens during the S stage of interphase, you can see the DNA just duplicated. The chromatin strands duplicated. And that happens in the S stage of interphase, which is prior to mitosis. Now, in the cell to the bottom, the same thing. The cell to the bottom is going to duplicate the strands of DNA. It's going to duplicate the chromatin. That happens during the S stage of interphase, which is prior to meiosis. So far, the two cells are setting up to be fairly similar to one another. Well, let's move on. Now we're going to move on to the step known as prophase. And let's look at the cell on top. It's going to go through the prophase stage of mitosis. And so what happens is the nucleus is going to dissolve, and you see the blue circle disappearing. The next thing that I want to note is look at the bottom cell. The bottom cell is also going to lose its nucleus as it goes through prophase number 1 of meiosis. In the top cell, what I want to draw your attention to in the top cell now is that the chromatin, the strands of chromatin that were just duplicated, are going to coil into chromosomes. I'll explain the P and the M in just a moment. In the bottom cell, prophase number 1 of meiosis, really the same thing is going to happen. The chromatin is going to coil into chromosomes. Now I want to mention what the P and what the M stand for. The P, this key is going to stay up here for a little bit. The P stands for chromosomes that are paternal. Paternal chromosomes are chromosomes you inherited from your father. The M stands for maternal. These are chromosomes you inherited from your mother. So notice how we have a chromosome number 1, maternal, and a chromosome number 1, paternal, M1 and P1. In both cells, there's also a P1, which means, in both cells, there's also a M2, which means chromosome number 2 from the mother. And there's also a P2, which means chromosome number 2 from the father. So M means maternal from the mother. P means paternal from the father. Well, let's move on with the process. So now that the chromosomes have formed, let's focus our attention on the bottom cell, prophase number 1 of meiosis. Something else is going to happen. There's a process called synapsis that's about to happen in the animation. This only happens in prophase number 1 of meiosis. Watch what happens. During the process called synapsis, the two homologous chromosomes are paired up with one another. The two chromosome number 1s are paired up with one another, the maternal chromosome number 1 and the paternal chromosome number 1. The same thing happens with chromosome number 2. The maternal and the paternal chromosome number 2s are paired up with one another. This pairing up of homologous chromosomes is called synapsis. This only happens during prophase number 1 of meiosis. And when synapsis is finished, this is what's created. These groupings of chromosomes here are called a tetrad. Tetra is a prefix that means 4, and the reason it's called a tetrad is because you see the bracket in the picture is around 2 chromosomes, but remember that one chromosome is made from 2 chromatids. So if there's 2 chromosomes in a tetrad, that's a total of 4 chromatids. That's how they get their name. This process called synapsis only occurs in prophase number 1 of meiosis. Okay, so the next thing I want to mention, now that synapsis has occurred, the next thing I want to mention is a process known as crossing over, and this only happens in prophase number 1 of meiosis. Watch this. In crossing over, parts of the chromosomes are going to swap with one another. Watch the animation. 3, 2, 1. Parts of the chromosomes swapped with one another. Parts of the maternal and the paternal chromosomes swapped with one another. If you missed it, here it is again on the other tetrad. So in crossing over, parts of the maternal chromosome number 1 break away and connect with the paternal chromosome number 1. So the maternal and the paternal break off parts of their chromosomes and they recombine with one another. This process called crossing over shuffles up the genetic gene combination just a little bit. And we'll see why that's relevant near the end. So now when we go on to the next stage called metaphase, on the top, let's look at metaphase of mitosis. In metaphase of mitosis, the 4 chromosomes in the animation are randomly pulled to the middle of the cell. Look at the bottom animation now. In metaphase number 1 of meiosis, those 2 tetrads are going to be pulled randomly to the middle of the cell. And that's really all that takes place. Remember, who's pulling the chromosomes here are those spindle fibers. And so let's move on to anaphase. So now at the top again, anaphase of mitosis. What happens next? Here it is. What happens next is the chromatids, the sister chromatids, are pulled apart from one another. The spindle fibers will pull one of the chromatids to the left, the other chromatid to the right. So the chromosome is ripped apart. One chromatid pulled to the left, one chromatid pulled to the right. Well, now what about the meiosis stage? Anaphase number 1 of meiosis. Watch this. The chromosomes are pulled apart. So the chromosomes are still intact. I should say the tetrads were pulled apart, but the chromosomes are still intact. 2 entire complete chromosomes were pulled to the right. 2 entire complete chromosomes were pulled to the left. So that's a bit of a distinction that's different between the mitosis on top and the meiosis on the bottom. Let's move on into telophase. So in the telophase part of this presentation, the process known as cytokinesis is going to start to split the cytoplasm of the cell, and the 2 cells are going to split in half. You know, sometimes cytokinesis starts during anaphase, but I just threw it in here in the telophase because this is kind of the final step. Telophase is the final step of mitosis. So cytokinesis is going to split the 1 cell into 2 when the cytoplasm divides. The next thing that's going to happen is around those 4 bits of DNA that you see, the nucleus is going to regrow. The nucleus offers protection for the DNA that's inside. And finally, the chromatids, those 4 chromatids are going to unwind back into the linear strands of chromatin, very similar to how we started this video. And that's really it. That's really the end result. But look at the bottom one. We're going to see the bottom one's not finished. First of all, in the telophase number 1 of meiosis, you just saw cytokinesis split the two cells in half. Well, what happens next? The nucleus regrows, and sometimes the nucleus does not regrow, but in this animation we're going to show you what happens when the nucleus does regrow. The nucleus regrows, and that's really it. The chromosomes are not going to unwind into chromatin, so that's really it, telophase number 1 of meiosis. But we're not quite done, because this was just telophase number 1. There's a round 2 of all this in just a moment for meiosis. So when we look at the top cell's finished. Mitosis is over. We started with we started with one cell with 4 strands of DNA, and we ended with 2 cells with 4 strands of DNA. But now let's look at the bottom. We can focus on the bottom now. Prophase number 2 of meiosis. There is no interphase number 2, just moves right on into prophase number 2. And like it says, the nucleus is going to often redissolve, and so there it goes. And when we continue on to metaphase number 2 of meiosis, the chromosomes are going to be pulled to the middle of the cell. Remember, who's pulling the chromosomes? That would be the spindle fibers. So the chromosomes are pulled to the middle of the cell. When we move on into anaphase number 2 of meiosis, now the chromatids are going to be pulled apart. So the chromatids get pulled to opposite ends of the cell. In this animation, one of the chromatids went to the top, the other chromatid went to the bottom. So the chromosomes that were lined up in the middle during metaphase, the chromosomes were ripped apart with one chromatid going each direction. And when we finally move into telophase number 2 of meiosis, well, the cytoplasm is going to split again during the process called cytokinesis, so the 2 cells have just split into 4. The nucleus is going to regrow, but now it's going to regrow in 4 cells. And the last thing that's going to happen here is the chromatids are going to unwind back into their linear strands of DNA called chromatin. And that's really the end of meiosis number 2 here. So when you look at the end result, what we finished with is 4 haploid cells. So just to wrap up this and kind of give this quick conclusion here, if you notice, I want to point your attention to the box in the upper left-hand corner. When we started this video, there were 4 strands of DNA in the one cell at the top. Well, let's count up the strands of DNA now. The new cells, the 2 new cells on top, look at that. 4 strands of DNA in the cell on the left and 4 strands of DNA in the cell on the right. So the cell started with 4 strands of DNA, and the 2 that are created each have 4 strands of DNA as well. And not only that, but both cells are identical. They each have 2 fully black strands of chromatin. They each have 2 fully red strands of chromatin. The cells are identical to one another. Well, now let's focus our attention to the one on the bottom. So the cells on the bottom just went through meiosis. But remember, at the start, they each had 4 strands of DNA in them. Well, let's count up the strands of DNA now. The new cells have half the amount of chromatin as they did from the start. Look at that. That cell only has 2. The cell in the upper right-hand corner only has 2 strands of chromatin. The cell in the lower left only has 2 strands of chromatin. The cell in the lower right only has 2 strands of chromatin. So the cell at the beginning had 4 strands of DNA, but these cells only have 2. Well, that's because these are haploid cells created by meiosis. Another thing I want to point out is that each cell is genetically unique. Each cell has a unique mixture of red and black strands of chromatin because the process called crossing over, which took place way back in prophase number 1. So that is one of the major unique differences of meiosis, which does not happen during mitosis. And this really will conclude this video right here. So the cells on top The cells on top started to go through went through mitosis. And so cells that go through mitosis will start diploid and end diploid. But the cells on the bottom, cells that will go through meiosis, will start diploid, but at the end, they're going to have half the amount of chromatin, and they're going to end haploid. So that's it. That's the updated video. I hope you like it better than the original one. I tried to be a little more thorough by adding crossing over. So I hope this was helpful, and good luck in your understanding of mitosis versus meiosis.